Hello and welcome. This is A&P Live Studio, and I am your host. As a registered nurse and anatomy and physiology instructor for advanced healthcare professionals, I'm here to guide you through some of the most intricate topics in medicine. Today, we're going to explore the complex world of Parkinson's disease, tracing its path all the way from a single misfolded protein to the profound clinical challenges it creates. So, let's begin where it matters most, with the patient. We'll start with a clinical scenario that many of you will likely encounter, grounding our entire discussion in the reality of patient care. I'd like you to meet Robert. He's 68, and up until recently, he was really enjoying his retirement. But his wife has started to notice some subtle changes, things that were concerning enough to bring him in to see a doctor. These are the classic, often quiet, first signs. A little tremor in one hand, but only when it's at rest. Maybe his face doesn't show as much emotion as it used to. He seems to be moving in slow motion, and his handwriting is getting tiny. These are the first clues to a puzzle that starts deep inside the brain. To really understand what's happening to Robert, we need to take a step back and appreciate the elegant system that controls all of our voluntary movements. Let's talk about the basal ganglia. Now, they don't actually start a movement. Instead, think of them as a sophisticated gatekeeper. They get movement plans from the cerebral cortex, and their job is to refine those plans, to filter them, to make sure our actions are smooth, purposeful, and controlled. And they do this using two key pathways: a go pathway that facilitates the movements we want to make, and a stop pathway that inhibits all the movements we don't want to make. In a healthy brain, these are in perfect balance, and the conductor of this whole orchestra is a neurotransmitter you've heard of, dopamine. It's produced in the substantia nigra, and it keeps the go signal strong and the stop signal in check, allowing for fluid motion. So, what could possibly disrupt this finely tuned balance in Parkinson's disease? Well, the problem doesn't start with the circuits. It starts much, much smaller, at the cellular and molecular level. The absolute core of the problem, the first domino to fall, begins with a single protein that starts to lose its way. This protein is called alpha-synuclein. Now, normally, it's a perfectly helpful part of our neurons, helping to regulate the release of neurotransmitters. But in Parkinson's, it undergoes a really toxic transformation. First, for reasons we're still figuring out, the protein misfolds. Then, these sticky misfolded proteins start clumping together, forming aggregates. These aggregates build up inside the neuron into what we call Lewy bodies, and they're toxic. They disrupt everything, eventually killing the very neurons that produce our dopamine. And here's the really critical part. This process is slow and silent. It's estimated that by the time a person like Robert notices that first tremor, 50 to 70% of those dopamine-producing neurons are already gone. At the cellular level, this creates a vicious cycle, a sort of toxic triad. The protein clumps damage the mitochondria, causing an energy crisis. This leads to massive oxidative stress, which in turn causes more proteins to misfold. And all this damage triggers neuroinflammation, which just adds more fuel to the fire. It's a self-perpetuating cycle of destruction that drives the disease forward. Now, while most cases of Parkinson's are sporadic, a small fraction are genetic. And studying these families has given us incredible insights. What we've learned is that these genes are often involved in mitochondrial health and cleaning up bad proteins, which really highlights just how central those processes are to protecting the brain. Okay, let's zoom back out. How does all this microscopic chaos, this molecular pathology, lead to the symptoms that we can actually see in a patient like Robert? Let's connect the dots. So here is the direct consequence. With less dopamine, the go pathway gets weaker and the stop pathway becomes dominant and overactive. The end result is that the basal ganglia are sending out way too many stop signals. The brain is generating the command to move, but the gatekeeper is basically stuck in the closed position. And from this one core problem, you can trace a direct line to each of the classic symptoms. Bradykinesia, that slowness Robert feels, is the stop signal overpowering the go. The rigidity in his muscles comes from that constant inhibitory tone. And that resting tremor, it emerges when the normal rhythmic communication between different motor centers is disrupted. And it's so important to remember Parkinson's is a multi-system disorder. That alpha-synuclein pathology isn't just in the dopamine system. It spreads to other areas, affecting other neurotransmitters. This explains the huge range of non-motor symptoms, things affecting mood, the cardiovascular system, digestion, sleep, which for many patients become even more disabling than the motor problems. So now that we have a solid grasp on the core problem, not enough dopamine leading to a circuit imbalance, we can talk logically about how we try to treat it. First off, how is it diagnosed? Well, there's no single blood test or scan that gives a definitive yes. The diagnosis is still clinical. It relies on a skilled practitioner doing a thorough neurological exam and recognizing those characteristic motor signs we've been talking about. The cornerstone of treatment for many, many years has been a medication called levodopa, or L-DOPA. The strategy behind it is actually quite straightforward. If the brain's dopamine factories are dying off, we can help the ones that are left by giving them more raw material. And here's how that works. You see, dopamine itself can't cross the blood-brain barrier, but levodopa can. So the patient takes a pill, levodopa gets into the brain, and the remaining dopamine neurons grab it and use an enzyme to convert it into fresh dopamine. This new supply temporarily rebalances the circuit and eases the motor symptoms. It's a brilliant way to pharmacologically compensate for the deficit. Now, for more advanced cases, when the medication's effects become unpredictable, there's another option: deep brain stimulation, or DBS. This involves implanting a very fine electrode into overactive parts of the basal ganglia. The electrode sends out high-frequency electrical pulses that essentially jam that excessive stop signal, helping to restore balance to the whole system. All right, let's put all the pieces together. Let's summarize the entire sequence, connecting the molecular events all the way up to the clinical picture and our treatment strategies. You can see the whole arc of the disease here. It begins silently, decades before, with that misfolded alpha-synuclein. This leads to neuron death, which causes the circuit imbalance in the basal ganglia. That imbalance eventually manifests as the clinical motor symptoms we can diagnose. And it's those symptoms that we then target with therapies, all aimed at restoring that lost dopamine. And this really brings us to the most crucial question driving all the research today. Instead of just replacing the dopamine at the end of the line, what if we could go back to the beginning? What if we could stop the alpha-synuclein from misfolding in the first place? This is the next frontier, moving beyond simply managing symptoms to actually slowing or even stopping the disease process itself. Stay pathocurious, and I will see you in the next episode. Please remember, this presentation is for educational purposes only and is not a substitute for professional medical advice.